sequences play in proteins and how amino acid changes in these regions may affect these proteins. So, interestingly, disordered proteins, or IDPs, are these unsequenced structures. And if you know how the classic lock and key model works, where we have this here, and then we have these structured proteins, and this one will fit in because it's has the structure to fit in, but these ones, they can't because they're not the correct structure. Now, unsequenced structures do not have this rigid shape and can instead fit where they please. So let's just imagine this squiggly line. This is our IDP, and it is able to fit in here simply because it doesn't have the rigid structure that other proteins have. And the reason for this is these amino acids right here. So these amino acids are charged, hydrophilic, and polar. They are disorder promoting in proteins, and if you changed any or most of these to any other set of amino acids that were hydrophobic or nonpolar, this structure would start to become more sequenced and would lose this flexible shape that allows it to fit freely into this area. And a great example of how IDPs are very useful is the adeno-associated virus, which is used for gene therapy. Now, for gene therapy, you want the virus to deliver information to the nucleus of the cell that you are infecting. So how this all works is this is our cell, and we have our nucleus right here. And we have our virus, it's AAV, and it's going to travel along here and be endiosed into this via endocytosis into the cell, and it's going to be wrapped around in these endosomes. So I'm just going to draw these quick. And it's stuck inside these right now. Now, on its structure, AAV has these regions called, they're the VP, and this is VP1, VP2, and VP3, and they're on a scale about 1 to 1 to 10. Now, on VP1, it has this area right here, which is the VP1U, or unique, and this is a disordered unstructured sequence. So what AAV will do is deploy this area, and this unstructured sequence contains both phospholipase 2 activity and nuclear localization signal, and because of this, it is able to break free and find the nucleus. So after it breaks free, it will want to find the nucleus. And a really interesting fact is that the NLS has an amino acid sequence of PARKRL. Now, if you notice right here, all except for L are on the unstructured amino acids that will promote the disorder. So this fits the description of an unsequenced structure. So this is a practical application for this protein structure, and it is very helpful